This report brought to you by the Richmond Club, where investors and high-growth companies meet.
Thanks, Jeffrey. Our next speaker is the Vice Chairman, President, and CEO of Oncolytics Biotech. Please welcome Dr. Brad Thompson.
Thank you very much for coming today, and thanks for the opportunity to readdress this group before. Because we both filed with the SEC and the relevant securities commissions here in Canada, I'd like to draw your attention to our forward-looking statements and recent filings with the SEC and sadly still 13 different bodies here in Canada. Okay, this is a quick overview. We're developing a somewhat different agent, which is actually based on a live agent, which is a virus, and we call it the oncolytic virus, strictly as a cancer therapeutic. We've conducted a large number of clinical studies, which has been largely directed towards figuring out how this agent works, which I think we've completed. And that's resulted in a database that's actually quite large. We have just under 1,200 patients treated now. We have a very good understanding of the safety profile. We have a very robust patent portfolio. We have over 400, and 400 plus because they keep issuing, and it's hard to keep track of them, honestly. Issued patents, and we continue to file. And in our area, which is somewhat unique is that we're actually manufacturing the product at a commercial scale already, and that's actually one of the most difficult things in these new live agents, is not actually whether they work or not, but whether you can actually make them. And finally, for cash and cash equivalents, we have just over 20 million of cash at the end of Q2, and just to give you the runway, that gives us cash until mid Q2 2018 with our current burn rate. This particular product, as I mentioned, is based on a virus. It's called the reovirus. It's not raining today, but tonight when it starts raining or snowing, and just to give some perspective, where I live up in the Rockies, we had our first major snowfall of the year, August 16th. So the thought that you've made it till middle of October without snow risk is quite nice. But it's a proprietary isolate that's a little different than the wild-type reovirus that all of you would get exposed to if you were walking around out in the rain tonight or tomorrow. We tested your blood serum. Every one of you in this room would have, we'll call seroconverted to this virus. You've all been exposed to this virus, but it doesn't cause a disease. It's in the normal state. It's actually absolutely safe. And we use most because it was waffle, but all of you have been exposed to this. Now, with respect to safety, we can deliver this product two different ways. You can inject it directly into a tumor, or you can actually do it intravenously. And most of our patients have been treated intravenously, and they come in as outpatients into the clinic. They sit down, they read a book, go to sleep, watch TV, put an IV bag on their arm, and half an hour later we send them home. And it's a very benign way of treating patients. And we have never reached what we call a maximum tolerated dose, which is, if you read your aspirin bottle or your Tylenol bottle, that daily dose maximum is what we call maximum tolerated dose. There is nothing for this. We've gone to extremely high dosages, much higher than we use in clinical trials, and we still don't know what the top end is because it's just that safe. If you use the product as a monotherapy, its safety profile is very consistent. On day 2 or day 3 of a 5-day treatment cycle, you will get a mild fever, almost guaranteed. About 90% of the patients have that mild fever. About half of you would feel low-grade fatigue, and if we took and some other minor side effects occasionally, and if we looked at your blood chemistry, some of your white blood cells would have exfiltrated into the tissue chasing the virus because it's an infection, and they come back later. So it's a very short-term depletion of your white blood cell population. It's a very, very predictable and very reproducible side effect profile. This agent works in two different ways, and this is more defined than the last time I had the opportunity of talking with this group. The first way is, based on genetics, we can actually predict whether this virus will actually kill tumor rapidly. And how it does that is by infecting tumor cells. If these genetic mutations are present, then the virus will replicate and kill that tumor cell. And usually within 2 or 3 days, patients are reporting to us already that they can sense differences in their tumors. They're softer, they're different, they're already smaller. And if you don't see a response within days and sometimes weeks at most, now you know you've missed the mark. And this is a critical point for us to be able to pre-screen patients, especially with first-line patients. The first time you treat a cancer patient is the person's best chance at a successful outcome. And if we can screen patients as if they've got that list, any one of those mutations, then with a very high degree of probability they're going to respond to this agent. So you treat them with it. If they don't have those mutations, put them on another agent so they have a better chance at actually having a response. And that supports premium pricing and premium use in markets like the United States and Western Europe. A secondary effect, which actually probably results more in lifespan changes, is the virus actually interacts with your immune system after we treat you for that first part. And I won't go into the detail on this because it would turn into a lecture, which I'm fond of giving, but I wouldn't bore you to death today. But the virus interacts with the immune system in two very specific ways. And actually, this is what results in lifespan extensions that we're seeing now in a number of randomized clinical studies. So if we look at studies where we're looking at what we call progression-free survival, which is just when your tumors start to grow again, they progress, so you're progression-free, and they measure that. And overall survival, which is the really important thing for patients. We do now have data out of a number of randomized clinical studies that support both of these. Now, one thing we reported this summer that is quite different is that we never actually confirmed in patients these genetics. We've been working on this agent for quite some time now, and all that assumption about the mutations was based on animal model data, which is quite common. And that's not good enough for the FDA. If you want to go into the FDA and say we're going to screen patients in a phase 3 study for these mutations, you have to have clinical supportive data to say that you're going to do that, so that the patients are deriving benefit. This was a randomized phase 2 study run here in Canada for a specific type of non-small cell lung cancer, which is non-squamous as opposed to squamous. And what we were comparing here is the patients with the mutations with the patients without the mutations. And this would confirm whether you were getting benefit based on that. So if you look at median progression-free survival, which was the primary endpoint, you're between triple and quadruple the PFS benefit by having the mutations as opposed to not. And you look at median overall survival, it's not quite triple, but it's just under tripling the lifespan if you have the mutations. So if you're a patient, you screen for this. If they have it, you can guarantee them a benefit over if they didn't have the mutation. And so the patients on the left there without mutations, in the future we would move them into other clinical studies or other treatment regimes. And the patients on the right are the only patients that we will treat going forward. This was absolute key data, and the FDA was extremely pleased for us to have this. This is just Kaplan-Meier curves, which we're all fond of showing. On day 0, 100% of the patients are either alive or haven't progressed, and when it hits the bottom, that means they've all progressed or they've all died. So a curve that's to the right is better than a curve that's to the left. So of course we handily put the one on the left in red to show you it's not as good. But the one on the right is the patients with the mutations, and the one on the left is without. And if I were a patient, I know which curve I would prefer to be on as a result of that. If you look at this study from its other perspective, and that was a pre-planned analysis, by the way, just so you all know, that last analysis was us doing something we put into the clinical study at the start. If you just look at the patients on the standard analysis, looking at control versus test, the control arm in these patients would have just got pemetrexed. The test arm would have got pemetrexed plus reolysin. Here you're looking at the true measure of efficacy in the general patient population. This is screened and unscreened. And what you see is you have a statistically significant improvement in median progression-free survival and a strong trend, not statistically significant, in overall survival. And so, again, patients are deriving both overall survival benefit and PFS benefit by having reolysin added to this particular population. This was the second indication that we've had that gender matters, by the way. If you look at the males in this population, they actually, and again, it was a predetermined analysis, the males didn't see nearly the benefit that the women did, which is interesting. And we saw that also in colorectal cancer, which a number of you have seen me talk about before. Again, Kaplan-Meier curves, I mean, progression-free survival is very clear that you derive benefit, and overall survival is a nice curve because all the patients derive benefit as opposed to the patients on the left on the curve beside it. So looking at tumor responses, those overall survival data we now believe is due to the immune system. And we'll be coming out with data later this year in multiple myeloma and 2 other indications that has much stronger detail based on immune interactions for those of you who follow that sort of stuff. The other area that we look at is tumor responses, because this agent is exquisitely good at causing patients to have tumor responses. And where we're focusing our attention on is something a little different. And again, you've heard me speak of this before. It's looking at metastatic disease. Now, if you look at an overall patient population that comes out with a cancer, so if we're mostly male or all male in this room, so prostate cancer is a gender-specific disease. If you're a male and you live long enough, you will get prostate cancer. That is death, taxes, and prostate cancer are unfortunately a commonality when you're a guy if you live long enough. But if you look at those patients who die of prostate cancer, they're not dying of the primary cancer in the prostate gland, the overwhelming majority of the time. They're dying when it spreads. It's the same with GBM, which we've talked about before. I mean, if GBM never spread, it wouldn't be an issue, but it does, and that's why it's so lethal. You could say the same thing for colorectal cancer, bladder cancer, breast cancer. I mean, if you can get it early and you can cut it out, then you can cure it. I mean, in colorectal cancer, I mean, if it's just in the colon, it's like a garden hose, literally. If you have a leak in your garden hose, you cut out 3 or 4 inches and you glue it back together again. That's what we do with colorectal cancer in surgery. And it works, unless it's spread, and then you're in deep difficulty. But 9 out of 10 cancer deaths are due to cancer when it spreads. And so, and there really aren't any agents specific to metastatic disease. So it's a large unmet need. Those metastases, as we call them, spread are largely in the liver, lung, lymph nodes, and the brain. And we've always thought that reolysin should work on metastases. And until recently, we didn't have any clinical evidence for that, but we do have that now. And the reason we thought that is where the virus goes when we put it in the body, which is biodistribution. It's selectively cleared in the liver, lung, and lymph nodes on the first pass. And of course, the question was, as we heard before, would get into the brain, and this is one of the agents that actually does get into the brain too. We demonstrated that in a separate clinical study in the UK. The virus does get across the blood-brain barrier. So all the places where metastasis is, this virus goes selectively. So it should work there. Then if you look at the genetics, and that's why it's key what I told you before about genetics, if you look at the genetics of metastasis, virtually all metastatic disease has one of those mutations. So basically, just by saying metastasis, you've already pre-screened a patient to success for metastatic disease treatment with this agent. So we always thought that was the case, but we had to prove it in the clinic, and we have. The first thing we did looking at liver metastasis, and the reason we're focusing on liver metastasis is because it causes the most deaths as a percentage of all those metastases. There's 1.2 million people a year in developed countries die of metastasis to the liver from somewhere else. And so we were looking at head and neck cancer patients who all had metastasis to the liver, and in single-arm studies, because they're only anecdotal because you don't have a control arm, our doctors were noticing very unusual, what they called unusual responses. But it is anecdotal, but they are interesting to look at. And again, you've seen this slide before, some of you. This was day 0. This is the liver. All these dark splotches are the liver metastasis. This patient was actually in an advanced state of liver failure and probably had days left to live. Post-cycle 2, which is only 3.5 weeks out, you can see virtually all the metastatic disease is resolved. We only treated these patients 8 cycles. I don't have that 8 cycle up there, but the response is maintained. So it's very, very common for this. And it doesn't seem to matter whether the primary tumor up in their head, they have a response or not. Sometimes they do, sometimes they don't. We almost always get liver responses though in these patients, which is interesting. So we went into a phase 2 study, again single arm, anecdotal, and you see the same things. Again, liver, this is a fist-sized tumor in this poor patient's liver, and again, just a few weeks out, it's gone, pretty much. Very typical. But we had to show it in a randomized clinical study. You have to. So we did. We were looking at a randomized study again in head and neck cancer patients, and we were combining reolysin, it's a carboplatin paclitax, also a platinum and a taxane. So it's carbotax plus or minus reolysin. The control arm got those 2 drugs, the test arm got reolysin added into it. If you look at their local disease, you got some shrinkage, but it wasn't statistically significant. If you looked at their metastatic disease, and this is often backwards to what you see, we actually saw an increased amount of shrinkage, but in this case it was statistically significant. So our first statistically significant randomized clinical data demonstrating metastatic disease, selectively showing metastatic disease regression. Quite important. We've now done this in colorectal cancer too, and I actually presented this data to you last time. It was fresh at that point. If you look at patients in this had metastatic colorectal cancer, most of them, and the control arm got FOLFOX6 Avastin, and the test arm had reolysin added into it. If you look at the just overall objective response rate, so the tumors overall, primary and metastatic disease, you had just missed statistical significance, but you saw about a 50% decrease or increase in response rate from 35 to 53. But if you look at their livers, you actually see a statistically significant outcome. So in these patients, again, and highly statistically significant, that the patients in their livers are seeing a response. So head and neck and colorectal cancer to the liver, the commonality being the liver. This was our first study that I mentioned earlier, that we saw this huge bias towards women responding over men. We looked at the overall response rate in men, it improved about 10%. So if we ran a very large clinical study, and we're talking probably 1,000 patients, we could statistically prove that if that was indeed the case. When you looked at women, just under tripling the response rate. So 300% as opposed to 10% increase. And that was, again, very statistically significant. And this is the most extreme case of a gender-specific response that I've ever seen between two genders getting the same disease. And you also look, when you look at the liver metastasis by itself, this is the most extreme case. Again, if you just look at the women's responses in livers, they're much better than the men's responses in livers as well, for reasons we can't fully explain, because it's not just the genetics. There's something else going on, and probably their immune system. So we have filed, and it's actually cleared for a phase 2 study in the United States, where we're looking at females because they respond better with the genetics, because that's important, and looking at the overall response rate as the primary endpoint, and the liver metastasis-specific response rate. The first patient on the study will be enrolled next month, and this is down at the primary sites at Montefiore in the Bronx, just north of the island, Manhattan. The reason we did it this way was to get the FDA to actually agree to us pre-screening by gender, which they're loath to do. And so we had to get them to agree to that, and then combining in with this pre-screening by genetics, which is all that other data that I showed you. And that's because we're doing two very important screenings here, and they typically don't let people do that unless they have very strong supportive evidence. As soon as that first patient is enrolled, we'll be actually filing to convert this from a phase 2 to a phase 3 study. So we'll be actually filing for the phase 3 study in hopefully early in December. So there'll be a couple of patients on this study enrolled, and then they'll roll seamlessly into a phase 3 study. That's the plan. Another disease that the virus should work on, and should is a loaded word in cancer therapy, because should has been said more times than we can count when it didn't. But it should have worked in multiple myeloma. Multiple myeloma is a non-solid tumor, and there's leukemias, lymphomas, and MM. And so you get cancerous cells of various types being produced generally in the bone marrow and a couple of other places that get excreted out and they float around. Multiple myeloma is actually a cancer of antibody-producing cells, so B cells for those of you who remember some immunology. So we thought it would work here because if you look at bone marrow, reolysin actually gets into the bone marrow, which is unusual for an agent that big, if you want to think of it. If this was a drug, this building would be the virus. I mean, they're big, so it wasn't obvious it would actually get into your bone marrow, but it does, and it infects the multiple myeloma cells very effectively. The current standard of care, which are proteasome inhibitors, actually work best with reolysin. Proteasome inhibitors and reolysin are the best drug combination we've ever encountered. So that's bortezomib and carfilzomib. And of course, the other standards of care are the IMiDs, which is our nice way of saying thalidomide, because my friends at Celgene don't like us to say thalidomide out loud, so we call them something else. And in this case, immune effects are paramount. We actually see best evidence of immune effects. And this is what you see in immunology, and I apologize for sticking this in because most of us in this room, including me, aren't immunologists. But the interesting thing here is the exclusion. REOLYSIN actually restores the activity that carfilzomib suppresses in the immune system. Carfilzomib is really horrible on your immune system. But if you look at CD8 cell populations, I mean, look at the left column and the right column. Carfi drops it, what's that, almost 8-fold, which is not good. And REOLYSIN not only restores it, it actually more than doubles it, and so on and so forth. The key exception is CD68 cells, and we have no explanation for that, why that one subcell population in the immune system just, Carfi doesn't suppress it and Reo doesn't enhance it. We have no idea why. But these are key measures in the immune system for those of you who follow immunology. Briefly, with manufacturing and intellectual property, as I mentioned, these agents are difficult to produce. There's half a dozen companies making viruses for treatment of cancer, different viruses. We don't view each other as being competitive because they all act differently and have different cancers as targets. But a commonality is lack of ease of manufacturing. We've been manufacturing it at the final commercial scale in a facility in Carlsbad, California for a number of years now. And we're actually doing our first vialing in a commercially approved for sale vialing facility in Finland right as we speak, and starting to stockpile for sale on the shelf versions of this particular drug. And that company that we're working with in California is Sigma Aldrich Fine Chemicals. So it's 10 years of work from my manufacturing group's life to reduce to 2 bullets and a vial and a screen. In our area, though, this is more important than clinical data, honestly. The assumption is once we get into the clinic that our products are going to work in the viral area, and they typically all seem to be working. This is the Achilles heel in our sector. So we've spent a lot of time, and I have to say a disturbingly large amount of money on looking at manufacturing. We've actually spent over $100 million on manufacturing development, which is a lot of money for a small company our size. Patent portfolio, we have an extremely robust portfolio, and we did the entire portfolio. There's over 400 patents issued, including our core ones in the States. There's 60 in the US and 20 Canadian, and we continue to file. Our composition of matter claims go to 2028. So we actually own this virus and the entire genus reovirus, which is all the family members as well. And we have all associated claims otherwise that go beyond that date. From a corporate financial perspective, our parent exchange is Toronto. We're on the foreign filers OTC, QX, and are fully SEC registrant. And we also trade on Frankfurt, though not willingly. They just listed this. We tried to get off, but that's not the way Frankfurt works, as we all know, but we feel it's appropriate to disclose. And as I mentioned, we have cash to take us till mid-Q2 2018 with our current burn rate. So just investment highlights. We have 4 randomized clinical data sets coming out in the next 6 months from different studies, 2 in Canada, 2 in the United States. We are actually preparing to file also our lead registration study in multiple myeloma. We don't consider the colorectal one as because we're already getting into it with the phase 2 in the States, though that will be the other phase 3 study. We have great safety data and a very strong safety database, a good IP portfolio, and I can't emphasize enough times how important that last bullet is, that we actually are already manufacturing this very difficult agent at commercial scale. So that's all I wanted to say today. I'd like to thank you again for your time and attention and the opportunity to present to you again. And I guess we have a couple of moments for questions.
This report brought to you by the Richmond Club, where investors and high-growth companies meet.